My name's S Lindell, and my current title is Senior Director of Product Management. I've been with Lineagen for close to 10 years, worked with Dr. Paul that entire time. I was introduced to him through the University Venture Fund, which is a student-managed venture fund that was spun out of the University of Utah, the first of its kind. Dr. Paul came to us for some financial analysis when Lineagen first got the assets from the state of Utah and from the University of Utah to try and figure out how best to commercialize those assets. So I led that project because I had a background in genetics and a business degree. After I did that project, I came on as an intern in between my first and second years of the MBA program, and then I stayed on full-time ever since then. I think that was in 2003. I think the great thing about Lineagen is the decades of research that we were able to build this company upon. The Utah Population Database, the people at the state of Utah, at the Huntsman Cancer Institute, Steve Prescott, Mike Levitt, John Huntsman, all of the politicians and the leadership at the University of Utah that made that technology available, that built that technology, and then gave us access to the technology to be able to do this type of research. So we really are able to build upon decades and decades of research. It was, a lot of that work was used in the Human Genome Project. All those Caucasian families came from Utah. Mark Leppert, one of our scientific co-founders, he was the one that really, generated all those Caucasian reference families for the Human Genome Project and then for the follow-on HapMap project. So again, we got to just build on all of that work that was done before that. And then since then, we've been able to build a really exceptional team because we have access to what is probably the best genetic resource in the world, is the Utah Population Database. So being able to access that, being able to access those thought leaders, Steve, who's the chairman of our board, Mark Leppert, who's the chief science advisor, and having access to those types of minds has really made a big difference for us. I think the relationship between Dr. Paul and myself is, I think, is very complementary. I think we have a lot of skill sets that play off one another really effectively. So he has vision, and he's able to kind of harness the high-level vision of these scientific luminaries, and Dr. Prescott and Dr. Leppert, and then I'm the detail guy, and I put it pen to paper and kind of make the business model and develop the tactical strategy and the plan. The thing that interested me initially was just the genetics, the puzzle, being able to, the problem, the massive, 3 billion bases, and the ability to use that information to kind of, decode these instructions of the human body. The ability to be able to do that was something that was really exciting to me. And I realized pretty quickly that the Utah Population Database was a really unique resource that you really weren't going to be able to access anywhere else in the world. I mean, this combination of these huge families in the Mormon population. I mean, 10 kids who each have 10 kids who each have 10 kids, and all of a sudden in just a few generations you have hundreds and hundreds and hundreds of people, and then they're exceptionally participatory. So our participation rates for genetic studies in Utah is like 90%, compared to Duke, where I think the researcher told me they're lucky to get 50%. And then having all of that information, the genealogy, the best genealogy in the world. You can go to countries in Asia and find really huge families, but you have no idea how people are related to one another, and being able to kind of trace lineage in those. I said it again.
Take it from and being able to trace.
And being able.
And look at Diane.
And being able to trace lineage in those big families in other countries is really impossible without that data source that's the Utah Population Database. So I recognized that pretty quickly when I started the project. But I think as we've developed the business model, we've started to offer it clinically, and you can see how patients use this information and how they change the way that the doctors are treating medicine. We're dealing with doctors that before we came along, they were afraid when they saw a kid with autism coming down the hallway. This is going to take forever. I'm going to have to give the parents bad news. I'm not going to be able to do anything for them. And now they're in a position where I think when they see those kids coming down the hall, they feel confident about their ability to be able to help that child, their ability to be able to tell the parents what they need to be able to tell the parents, and do it with confidence and do it in a way that they don't feel like they're doing a disservice to the patient by even opening their mouth. So I think, as we've built the company and as we've got out there clinically, you really get to see the utility of this in clinical practice, and that has been exceptionally exciting for me. Although I still get to play with the science at a pretty deep level when I want to. The generic name is microarray. So gene chip is a registered trademark of a type of microarray. And there's a bunch of different microarray technology that's available. The Affymetrix array that we use is Affymetrix is the pioneer in microarray technology. And the chip that we use from them today is the highest resolution chromosomal microarray available for the type of testing that we do. And we have partnered with them to design a custom microarray. And I'll explain exactly what that means in more detail in the slides. So the technology we use is called microarray technology, or chromosomal microarray technology. The acronym for that is CMA. The registered trademark of the brand that we use is the gene chip. It's manufactured by a company called Affymetrix. We've partnered with them to develop a custom version of this microarray that has the highest diagnostic yield, or the highest resolution for what we're doing. If you think about it in terms of digital cameras, we're using the highest resolution digital camera that's possibly available. So there's a lot of point and shoot and disposable cameras that are out there, but what we're using is the best digital camera that you could buy, and that's what this chip represents. The technology that goes into this chip is probably the coolest technology ever developed. It's unbelievable if you think about what they have been able to do. So to make this technology work, they had to create silicon chips, just like the computer chips in your computers. And then what they've done is they've learned how to synthetically create human DNA in the sequence that they want that DNA in. And then they learned how to adhere that DNA to that silicon chip. And that's just what's in this piece. Then you have to have the analysis technology and the software to be able to analyze and evaluate what the chip is telling you. So the amount of science that went into this, the silicon work from computer scientists, the synthetic biology from biologists, and then the adhesion chemistry to get it to attach, it's years and years of technology that's gone into this, and Affymetrix is really the pioneer in developing that technology. So that's why we're excited to partner with them to build this custom array that we're using. So the slides that I prepared kind of talk about this, what happens in this chip at a more micro level. So in the first slide, what you see is it's an array. It's a bunch of small features. And within each one of those features are millions and millions of single-stranded DNA. So if you think of what DNA is in the human body, each human has DNA, which is the instructions for what we look like and how we behave, and all the different traits that make people different from people and people different from animals. In that DNA, each person has 3 billion single nucleotide bases. Those are the building blocks of DNA. And what we're testing with this array is looking at one of those DNA nucleotides every 1,000 or so across the entire genome. So we're looking at about 3 million total DNA nucleotides with this technology, and that represents a really high resolution image into what you're looking for in the DNA. To give you a reference, for forensic testing, they only look at 13 DNA markers. And that's all that's really needed to be able to tell one human apart from another. So we're looking at 3 million. It's a very high resolution analysis, and what we're looking for are big chunks of DNA that are missing or are duplicated. So for whatever reason, the machinery that created the DNA made a mistake and it duplicated it or just got rid of big chunks of DNA. When I say big chunks of DNA, we're looking at things maybe on the order of tens of thousands of bases to millions of bases that are missing. Down syndrome, for instance, is a whole chromosome that's been added in there, and that's what causes Down syndrome, is an extra chromosome of a few million bases that have been added to a person with Down syndrome. So we're looking for things that are smaller and in some cases even bigger than that. So the way that you do that is you create this synthetic DNA and you put that on a chip that's shown in this slide. So what Affymetrix does is they create these single-stranded pieces of DNA called DNA probes, and then they attach those to the silicon substrate. And they put those on the array, again, millions and millions and millions of these DNA strands in known sequence, and they put those in known locations on the array. So what they have is an array that has features that measure these 3 million different locations across the human DNA. And they know exactly where it is on the microarray. So the software can, when the software goes to do the analysis, it knows exactly what's supposed to be there and what they're looking for. So they take that, and then what they do is they put human DNA, the human DNA sample from our patient, on top of those single-stranded DNA. And each one of those human DNA fragments has a little fluorescent tag on it. And so if it binds to the single-stranded DNA probe on the array, then it'll light up. And since you have millions of DNA strands for each one of the markers that you're looking at, what the technology actually does is it measures the intensity of those fluorescent tags within that feature, which is looking for just that one location. So, sorry, I skipped ahead to slide 2. And, I think it'll be pretty obvious when you look at the slides, yeah. Okay, so I'm just going to go then to slide 4, because I think I kind of that explanation takes you through slide 1, 2, and 3. So in this slide, what we're showing is the raw output. And the raw output just looks like a bunch of different colored dots. And it's not something that you could read by eye. So the other piece of this technology, which is phenomenal, is the laser technology that's used to measure the fluorescence and how intense the fluorescence is that's coming off the array, and then the software that analyzes exactly what that means. So if it's very bright, that means it bound 100% and that DNA exists and it's there as you would expect. If it's lower intensity, that might indicate that it's partially bound, but not like what you would expect. So maybe there's not enough DNA, or the DNA is just slightly off. And the output that we get then off the software tells us, here's the places where there's DNA missing and where there's DNA added in these big chunks. And that is being compared to DNA from thousands and thousands of people that have been used to build that reference set. So what we get is an output of every big chunk of DNA that's either missing or duplicated. And then what we do is we investigate very thoroughly exactly what that means. Is it hitting a gene? Has that gene been implicated in autism, developmental delay, another type of neurological function? Is it a gene that's been discovered but that we don't really know what its function is? And then we scour the literature to find out, what does this mean? Is this what could be causing the patient's symptoms? And that takes an awful long time to do that work. And we take great care. Our genetic counselors and our cytogeneticists spend an awful lot of time looking at every single one of these changes that we see in each person to try and determine if that's what's causing their disorder. And if we determine that it is what's causing the disorder, in many cases we're changing the way that these patients are treated. And we have a lot of kids with a diagnosis of autism because that's how they present clinically to the physician. But in reality, what they have is a well-known genetic syndrome that just has a bunch of overlapping symptoms. If you think about what's going on here, there's 700 disorders of childhood development that have a genetic basis. But we're talking roughly 30, 40 symptoms that they all share. So there's a huge imbalance there between the number of disorders and the number of symptoms. So if you're a doctor trying to make a differential diagnosis and use a rule-out type of a methodology, you'll never get there with using symptoms alone. You just won't get the diagnosis right, and it happens time and time again. And so you end up with these kids that have these kind of placeholder diagnoses, right? Developmental delay. I mean, that's not really a disorder. You're just describing the symptoms that a kid's not meeting their developmental milestones. So a genetic diagnosis is an objective assessment of what's really going on and what's happening with the underlying biology. It moves you from, as a clinician, from treating symptoms to treating the underlying biology, what's actually happening at the patient's level. We have one example of a kid who had a diagnosis of autism spectrum disorder. He had been in intensive speech and language therapy. His parents described temper tantrums because he couldn't communicate with them what he wanted. So he'd just kind of, throw a tantrum and scream and yell multiple times a day. And then when he got the diagnosis, what he really had was Angelman syndrome, which is a dramatically different disorder than autism spectrum. And one thing that's a hallmark of Angelman is that these kids, they just don't develop speech and language at more than 10 or 15 words in a lifetime. And so what they did is they took him out of speech and language therapy, put him in alternative communication therapies, using pictures to communicate, using sign language, and he's excelling in those types of therapies. And he had been languishing in that speech and language therapy for a couple years. And he went from being able to have almost no communication with his parents that was very effective in either direction, to being able to communicate with them on a regular basis, almost everything that he needed. Instead of having to scream and yell if he wanted a glass of milk, he could just go and grab the picture of a glass of milk and hand it to his parents, and there was no tension there. And so he went from having a few outbursts a day to having maybe one or two a week. And so if you can imagine how serious of a change that is for these families. That's just one example, and we've tested over 1,000 kids now. So what we're doing to help these kids and to help their families and to change the way that their doctors practice medicine, the way these kids are treated, the way these kids are treated at home and at school and in life, and I think really preparing their parents to help them throughout their lifetime. Whereas until you have a diagnosis where you're really confident in that diagnosis, it's really hard to plan for the future. And what can you even expect, right? I mean, I think that in that case that I presented, they had the improper expectations. They thought that their son was going to be able to talk and communicate in sentences and paragraphs, and that just wasn't the case. And so it reset their expectations of what they can plan for in the future. I think that's called anticipatory guidance. Really, what can I anticipate? Are they going to meet these cognitive milestones? Are they going to go to college? Are they going to be able to live on their own? Are they going to be able to have a job? Do I need to set up a living trust for them? And all these types of questions that they come in a flood when you get this initial diagnosis. And a lot of parents are making those types of decisions about the long-term future for their kids, and it's based upon a wrong diagnosis, because it's really not possible to make this diagnosis unless you have some objective measurement. And genetics is the best tool we have to do that today. So I think what sets Lineagen apart from other companies that do genetic testing for kids with autism and developmental delay, and there's plenty out there, is that we are driving the biological understanding of autism and developmental delay. What these other companies are done, they either laboratories or they're technology companies. So they've invested in a laboratory, and what they're interested in is best utilizing that laboratory. Or a technology company that's made a type of microarray like this, that's looking to use that microarray in as many places as they can. So they use it for autism, they use it for cancer, and they're really experts in the technology, but not necessarily experts in the disorder. And if you're a laboratory, you're an expert at running a laboratory, you're not necessarily an expert at the disease. We don't have a laboratory, we don't develop the technology ourselves. What we do is we partner with the best laboratories and the best technology manufacturers. So we can always stay at the very leading edge of the technology and of laboratory science. Where we put our proprietary investment is understanding the disorder, understanding the genetics of the disease, understanding that underlying biology, so that when we get the results from the technology and out of the lab, we are the best company to know exactly what you should do with that, what it means. The proprietary array that we're launching, the second generation of First Step DX, represents almost a doubling increase in the diagnostic yield of any other laboratory test that's out there for autism and developmental delay. So right now the diagnostic yield is 15 to 20%, and that's about where our competitors will remain And with this proprietary technology that we'll launch, the custom array that we developed in partnership with Affymetrix, will boost that diagnostic yield to close to 40%. And that means we'll double the number of kids that we can provide that objective diagnosis to. And I think there's just no way to compete with that. I think since we are doing the research, since we're driving that diagnostic yield, we can immediately make it clinically available, whereas our competitors have to wait for us to publish those results and wait for the patents to run out before they can even think about being able to use that technology and being able to run those tests. So what makes Lineagen special is that we do research, and we do research focused on autism and developmental delay, and then we partner with the technology and the laboratory companies that are the best in class. What we think is happening in autism is this. Have you heard of the triune brain theory?
No.
You have three parts of your brain. You have the middle brain, the reptilian brain that controls your fight or flight response, and then you have the middle brain, which is your mammalian brain. It's your emotional brain, your emotional reaction to situations, and then you have the outer brain, which is your human brain, the thinking brain. That does deductive reasoning and language, things that other animals don't have. And what they believe is, if you think about social interaction in a human context, it's probably the most challenging things we have to do as humans. You have to engage all three parts of those brains simultaneously. So when you meet somebody new, you have to decide, is this friend or foe, fight or flight? And then you have to have an emotional reaction at some level to that person, no matter what the situation is. And then you have to think. You have to have a conversation. You have to have language, and you need to have all those things working together. And what they found in kids with autism is, while they have as many neurons as other humans, they don't seem to build these long connections between the neurons and the different parts of their brain. You think about the stereotypical behavior. It's almost like they're stuck in one part of their brain. And so up until the age of 6, you have what's called this intense period of neuroplasticity, where your brain is developing at a really rapid rate biologically. Neurons are building these connections out from their central location to other parts of the brain. You're seeing pretty major chemical changes in the brain. I think so what happens with applied behavioral analysis, which is the most commonly used treatment for autism, is it's really intensive one-on-one therapy with a therapist. And what the therapist is trying to get you to do is engage those different parts of your brain simultaneously. And if you can start that therapy during that period of neuroplasticity, before they're 4 years old, and you do it intensively for 2 or 3 years, you can actually force those connections to happen, because their brain's still developing. So you're almost training the brain and forcing them. So what we've seen is 10 to 25% of kids actually are off the spectrum after 3 or 4 years of treatment. So what we need to be able to do is diagnose those kids earlier. That's still the average age of diagnosis is 5 years old. That hasn't changed for 20 years. So all this awareness and everything we've done, ultimately we've made no real change in terms of the age of diagnosis. And I think it's because it's a really hard thing to diagnose. So that's why applying genetics to that diagnosis is really critical, but you have to do it early. You got to do it before they're 4 years old, and that means it's going to have to be done by pediatricians who frankly just don't have the genetics training to be able to offer it. So that's where Lineagen comes into play. We have genetic counselors, we have pediatric geneticists, we understand the technology with molecular scientists and partnerships with companies like Affymetrix, so we can bring that technology to bear on those frontline pediatricians that are the ones that really are going to be responsible for changing this whole diagnostic paradigm, getting these kids diagnosed early, getting them the treatment as quickly as possible, and then providing them with a medical home as they go through that treatment, so that we can get more and more kids off of that spectrum. Because I think what we can learn from kids with autism, I've met a few kids with autism that were autistic and didn't have communication ability, but then they went through this treatment and now they're able to communicate. The way their brains work are so different than the way that my brain works, that when you meet somebody that has that different type of brain functionality and can explain what they're thinking and their thought process, I think what we can learn from them in understanding human intellect and thought processing is going to be, it's going to change the way that other humans function, and that other humans think about their health and their cognition and personality and social interaction and behavior. And so getting these kids to a point where they can be able to explain that when they're adults is really going to change life for everybody.